Hi everyone, my name is Reagan Brummett, and in this presentation I will show you my poster about how our cells can turn simple things like glucose and carbon dioxide into the chemicals that DNA needs to make copies of itself. Okay, we're going to start off with block 1. Okay, this process starts with the production of glucose 6-phosphate. This is the first step in the process that will be carried out. It eventually makes it to the pentose phosphate pathway's oxidative phase, where it produces ribulose 5-phosphate and NADPH. In order to construct nucleotides, it is subsequently changed into PRPP, which stands for the sugar backbone. Next, for my block 2, we'll talk about galactose to glucose 6-phosphate. This sugar is created when the lactose in our food is broken down. The conversion of galactose into glucose 6-phosphate, the same molecule discovered in my block 1, is brought about by the Lalor route. Next, we're going into block 3, which is the lipid breakdown from ketone bodies. So the lipids also, they will play an important role in this. Glycerol, the structural element triglycerides, is also produced in the glycolysis process, along with NADH and FADH2. When there is a limited supply of carbohydrates, acetyl-CoA builds up and it is subsequently transformed into ketone bodies like acetoacetate and beta-hydroxybutyrate. This illustrates how lipids also aid in the synthesis of ATP. And now we're going to go on to block 4, which is the purine and pyrimidine synthesis. It is now possible to use the PRPP we made to build nucleotides. In the process of making pyrimidine, PRPP and ORate come together to make UMP, which eventually turns into CTP and TTP. These processes use a lot of energy and shows how important the nucleotide synthesis for the cell to work properly. On to block 5, which is nucleotide activation. For block 5, which is nucleotide activation, to use nucleotides, they must first be activated into triphosphate forms. For example, UMP can be changed into UTP and can then be changed into CTP or TTP. The purine side of the cell turns IMP into AMP or GMP. Then those AMPs and GMPs are further phosphorylated to make ATP and GTP. These high-energy forms of nucleotides are used in both DNA replication and RNA transcription. Next, for our block 6, it is ATP synthase and chemiomosis. This region makes more than half our ATP. The ATP synthase enzymes moves protons that were pumped into space between the mitochondria's membrane back into the matrix. And next we're going to move right on to block 7, which is the sucrose breakdown of glycolysis. Now let's talk about glucose and fructose again. Fructose comes from sucrose. Glycolysis then happens to both sugars, and this makes pyruvate. Pyruvate is an important part of how the body breaks down energy, and we'll be talking about this more within the next blocks. Block 8, which is glucogenesis, is essentially the opposite of glycolysis. When the body needs glucose, it can make it from things that aren't carbohydrates, like glycerol or pyruvate. Now we're going to go into block 9, which is this block, which is lipid biosynthesis. Focus on how fat is stored and how cholesterol is made. When glycerol 3-phosphate and fatty acids come together, they make triglycerols. Acetyl-CoA also helps mevalonate pathway, which eventually makes cholesterol. Making mevalonate from HMG-CoA is the most important step. Now moving into block 10, amino acid synthesis block. This is right here. Other intermediates from glycolysis in the TCA cycle can be utilized to make amino acids. For instance, one potential result of the 3-phosphoglycerate is a conversion of serine to glycine. Tyrosine is one of the building blocks for neurotransmitters and can be created by converting phenylalanine. Thus, metabolism supplies the building blocks required for the synthesis of proteins and signaling molecules, in addition to producing energy. Now we're going to move into block 11, the electron transport chain, which is this block. This is the process by which NADH and FADH2 provide electrons to the electron transport chain, located in the mitochondria's inner membrane. This establishes the proton gradient used by the ATP synthase. The final electronic acceptor and the building block of water is oxygen. We are now going to move on to block 12, which is the citric acid cycle. So for block 12, after the glycolysis is done, pyruvate turns into acetyl-CoA. Once acetyl-CoA is made, it joins the citric acid cycle. In addition to 3 NADH and 1 FADH2. In either 1 ATP and 1 GTP, carbon dioxide is made during each round of the cycle. And now we're going to move on to block 13, which is transamination. Transamination changes the intermediates of the TCA cycle into amino acids. Pyruvate turns into alanine, oxaloate acetate into aspartate, and alpha ketoglutarate into glutamate through the process of oxidation. Now we're going to move on to block 14, which is the cholesterol synthesis. The mevalonate pathway, which contributes 5 small carbon units, is continued in the formation of squalene. Squalene undergoes a series of changes to become lenesterol and eventually cholesterol. Now we're going to move into the last block, which is right here. And this block is the methionine cycle and succinyl-CoA. So methionine is transformed into SAM, which is a methyl group that controls DNA and proteins by the methionine cycle. SAM transforms into homocysteine, which can be recycled or used to make alpha ketobutyrate and cysteine. Because of this, the production of energy is closely linked to the metabolism of amino acids. In conclusion, metabolism is an intricate process that transforms amino acids, lipids, and glucose into components necessary for life. My poster demonstrates how these processes work together, recycle intermediates, and support the synthesis of proteins, DNA, and cellular respiration. Thank you all for listening to my poster presentation, and I hope you have a great day.